Hi students, and welcome to today's video. In today's video, I'm going to be going through with you the stages of meiosis, and we're going to be looking at each of those individual stages and drawing them and labeling them as we go. So if you don't already, I'd like you to get a piece of paper and first make sure that these notes that are right here in front of you are copied down with each of the stages, or at least make sure that you have access to them and maybe take a picture of it. And then make sure you have a piece of paper, and I'm going to show you exactly how I want that piece of paper set up right now. So here we have a piece of paper, and I have divided this into 8 sections. So I folded it in half and in half again one way. And then I folded it in half once again the other way. And when I unfold it, I have boxes. These boxes I then separated, and I'm calling prophase 1, metaphase 1, anaphase 1, telophase, and cytokinesis. And then we'll swing down and we'll go through prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. And so we're going to be following our path along each of these as we go. And then, so we'll be zooming in on like one phase at a time. And then once we have that phase drawn and done, we'll move on to the next phase. And at the end, we'll have this really nice diagram that explains all the important things about each stage in meiosis. So to start us off, we're going to be zooming in on prophase 1. So the whole point of meiosis is that we're taking a diploid cell and we're going to be reducing its number from diploid to haploid. So we're going to start out with a diploid cell. So we have a diploid cell, it's 2n equals 4. That means that we should have 4 chromosomes in here. But more importantly, because we're in prophase 1, we're going to have 4 sets of sister chromatids. Remember, we count the chromosomes by the telomeres that are present. So I'm going to draw my 4 chromosomes. So I'm going to have one set of chromosomes that is orange and another set of chromosomes that is blue. And they're also matched up by color. Now, this is one of those things where teachers, people who diagram this all over the place, there is a big problem with showing this part of crossing over, because on one hand, we need to show you that these orange chromosomes go together. But on another hand, I'm going to be showing you that these orange chromosomes are different. Remember, we talked about the idea that on the same chromosome, you could have one allele for blue eyes on that chromosome, and on a different version of the same chromosome, you could have an allele for brown eyes. And so those are the chromosomes we are lining up here. These chromosomes code for the same things, but they don't necessarily have the same alleles on them. Perhaps this one could be for brown eyes and this one could be for blue eyes. The way I'm going to show that is one of my chromosomes is going to be striped, and another one of my chromosomes is going to be empty or blank. Filled in the X's, one of each side. And what's important here to note is that it really doesn't matter whether the filled in or striped one is on the left or the right of any pair. It's completely random. But the pairs have formed, and now they're going to form something called a tetrad. So I'm going to look at the next slide here to give you an idea of what the tetrad looks like. So tetra means 4, and so our X's are going to be overlapping each other in 2 spots. Typically, I think of standing like with your arms and legs out, and imagine that you crossed someone else at the elbow and the knee. And now your 2 arms that we're crossing swapped. So now you have their hand and they have your hand. That is the idea of crossing over. And so what happens in crossing over is that our tetrad forms, and the tetrad is going to swap chunks of their DNA. So looking back here, we would have this overlapping moment here, and an overlapping moment down here. Same thing on the blue chromosome. And those would switch places. So we're going to redraw them in switched places. What I've done now in this crossing over is I've created this tetrad and I have swapped places so that I don't have any two chromosomes that are the same. So originally these two that were of the open form were the same traits, but they are no longer the same traits. So they have undergone crossing over. So in prophase 1, we see the formation of the tetrad and we see crossing over. So our two major points of prophase 1 are the tetrad and crossing over. Now it's worth it to note that in prophase 1, the other prophase kind of things that we remember from mitosis would happen. So the nuclear envelope would go away and the spindle fibers forming. Another thing that's important to note is that we are diploid at this stage and our pairs are of sister chromatids. So in this phase, we would say that we have homologous pairs of sister chromatids. So all of those things are a part of what is happening in prophase. So I should put in my spindle fibers forming and we're getting ready. Now at metaphase, these homologous pairs of sister chromatids are going to line up at the metaphase plate. So in this phase of meiosis, the pairs, which could be spread all throughout the cell, have now lined up exactly perfect at the equatorial metaphase plate. So we have our homologous pairs of sister chromatids lined up at the metaphase plate. And now in anaphase, much like in mitosis, in anaphase, these start to move away. But what is moving away at this point are the homologous pairs of sister chromatids. And so in anaphase, at the metaphase-anaphase transition, we begin to become haploid. So in metaphase, we still have a diploid cell. As we move to anaphase, remember that in anaphase, we move away. In this phase of meiosis, homologous pairs of sister chromatids separate. And so we are forming haploid cells. As we move through telophase and cytokinesis, we make two separate haploid cells. So through telophase and cytokinesis, we create two haploid daughter cells. Each has one set of sister chromatids. So each one of these ends up with one set of sister chromatids. And then we're ready to start prophase 2. So in telophase and cytokinesis, we're going to see the cellular machinery redivided. We're going to see the spindle fibers dissolve and the nucleus reform. And then as we move to prophase 2, we start the whole thing over again, but now with two sets of cells. So in prophase 2, both sets of cells start to attach to spindle fibers. So we see in prophase 2, the spindle fibers reattach, and this is going to look a lot more like mitosis. However, these cells are haploid. N equals 2 haploid cells. So we are already haploid, but we have haploid sets of sister chromatids. In metaphase 2, the sister chromatids line up at the metaphase. So once again, we see the chromosomes lining up at an equatorial plate. This time, though, it is our sister chromatids that are lining up at the equatorial plate. And now, in anaphase, our sister chromatids will break apart and be pulled away from one another into separate cells. Remember, anaphase still tells us that we're moving away, but this time we're separating sister chromatids. I think it's important to note that the cells are still haploid. And finally, in our last stage of telophase 2 and cytokinesis, we end up with 4 daughter cells, each unique in their composition of chromosomes. So by the end of cytokinesis, I have made 4 completely unique haploid daughter cells. And the reason why we say they're completely unique has actually everything to do with crossing over. When we crossed over these chromosomes, we made it so that no 2 cells would be alike when we were done. We have an orange open with a blue stripe. We have a top half striped orange with a bottom half striped blue. We have a bottom half striped orange with a top half striped blue, and a striped orange with an open blue. This means that all 4 of these offspring are unique. This gives us a whole lot of possible variation. And please remember that variation is the point of meiosis. And to put it all back together, in prophase 1, the homologous chromosomes join together at the tetrad and crossing over occurs. In metaphase, the homologous chromosomes line up at the metaphase plate. In anaphase and telophase, they are separated, and cytokinesis makes 2 haploid daughter cells, each with 1 set of sister chromatids. That is when we begin prophase 2, which is where our daughter cells attach spindle fibers and our sister chromatids now line up at the metaphase plate. In anaphase, sister chromatids are separated. Telophase and cytokinesis leave us with a total of 4 unique haploid daughter cells with single unique copies of each chromosome. And this gives us the diversity necessary for sexual reproduction to have a benefit to the organisms as a whole. The process of meiosis, detailed as it is, makes it possible for life on Earth to sustain the changes that occur within our environment, that have occurred, that are occurring, and that will occur in the future. Feels like it was really, really specific, but that's actually just the beginning. And as you continue more and more into biology and into higher levels of biology, you will learn more and more about what is happening in each phase of this meiotic event. But for now, in a nutshell, that will cover how we create our 4 unique individual haploid daughter cells from our somatic line in order to have reproduction. All right, you guys, I will see you in class tomorrow.